Science didn't stop there. Science then took this world of genes and went into the idea of, well, what about if we scientists can do stuff with genes? What about if we can change, block a regulatory gene, make sure genes aren't turned off? Or what if we can block the turning off of genes and make sure they stay on, and do all these things? And then they started to think about DNA and the whole idea that DNA is universal. And this idea that the universal nature of DNA means that any organism can read any DNA and form the same proteins. This concept that DNA is made up of the same triplets. And so a triplet codes for the same amino acid in all organisms. And therefore, by researching bacteria, which are some of our simplest organisms and easy to research because they're easy to grow, they're tiny, you can have millions of them, and there's not a lot of ethical problems in dealing with bacteria and maybe potentially destroying them. So in understanding bacterial DNA, we're understanding human DNA as well because of this universal nature. And by finding out, for example, in transcription that RNA polymerase is the enzyme that is needed to transcribe from DNA into a messenger RNA, that's the same in all organisms. RNA polymerase is RNA polymerase. Human mRNA polymerase is exactly the same as dog RNA polymerase, is exactly the same as plant RNA polymerase. So this idea of the universal nature of DNA, there's also a universal nature of enzymes that work around and deal with DNA. And this concept is what brought scientists to trying to maybe try things on bacteria and then extend it and see if that trial and error stuff works on other eukaryotic organisms. Till eventually we've got to the work of DNA manipulation. Now, in this section, and this is the second part of the area of Study 1, Unit 3, this section I see as two parts. There's the tools and techniques. There's the 4 key tools, if you like, or 3 key tools, and then what scientists do with those tools, what they use them for, which is the second part, the uses of DNA manipulation. And the world of DNA manipulation is often referred to as genetic engineering. And this is changing in any way or manipulating in any way an organism's DNA. And there are lots of ways it can be done. Genes can be introduced into an organism's DNA that don't normally exist there. Genes can be what we call turned off, silenced, if you like. Genes can be turned on when they're not normally. So there's a whole lot of gene editing that can be done, and there's a whole lot of introducing and removing genes. Any manipulation is called genetic engineering. Our course particularly centers on a form of genetic engineering called recombinant DNA technology. This involves the use of transferring genes from one organism into another organism. You may have heard of genetic modification. That is an example of recombinant technology. So this is the one we particularly want you to know the tools and techniques of it. And then we go into the world of how is it actually done? What's the outcome of these sort of things? And the outcome is going to be a whole lot of different outcomes. We can have organisms with new genes inserted in them for our purpose. We're not trying to create monster species, But all of this, we're hoping, is done to improve the food production of the world, medicines to help people live longer, get better, cures, all these sorts of things. So there's a whole lot of things. For example, it's used to make human insulin for people with type 1 diabetes, who up until now have had to use insulin injections, but the insulin's extracted from the pancreas of animals, in particular pigs. And that's what they're injecting. It worked, but surely it's better if they have human insulin, but not made from humans, not extracted from humans, done in a genetic modified way in using bacteria. DNA is universal. Introducing pest resistance into crops so that we don't have to spray with pesticides, and that goes into the waterways and also kills all our native bees and all this. So using those sort of things as well. Also, this type of thing is being used in cancer research, where we can actually, and it sounds awful, but we can actually induce gene mutations so that scientists can then try to find cures and ways to treat those different mutations, which in humans are different cancers. So the reason we're doing it is for everything we hope is proper good biology to improve society and human existence. So we're going to go through the tools, and then we'll look at the general techniques that this has been used for. And when we look at that, this is where we do have to just have an appreciation. It's not always, oh yeah, just go ahead and do it. There are ethical issues that need to be looked at. There are biological issues, and there are society issues that we need to look at in these technologies. All right. So what I thought we'd do is we'll just make a start, and we'll have a break in about, after I've done this first of the tools, we'll have a little break and then get back into it. Now, to do the whole world of, and we're going to be talking about recombinant technology in particular, what scientists have to do is, first of all, we have to find the gene of interest. For example, the insulin gene. You have to find it and isolate it. So if you want a human insulin gene, you need to get human DNA, and you need to get out of this mass of DNA, the genome, the insulin gene. So that's the isolation process. And there are tools involved in that, and they're the main tools. Once you've got, so this is where our tools work at to get that particular gene. Once you've got the gene, then genes sitting in a test tube do nothing. Genes only function if they're in a cell. So they can be transcribed and translated into the protein they code for, because it's the protein, remember, the insulin, that does the action. So what has to happen then, once you've got your gene, your target gene, you have to make sure you've got a way of transferring it into the cell you want it to be in. And then once that cell has received the target gene, in other words, we say it's been transformed, then we have to make sure that we have that organism functioning and not impacting on the rest of society, etc. So there are lots of steps involved in this process. So let us just focus first of all on this first one, getting the gene. Now, there are lots of ways to get cells. For example, if we want the human insulin gene, that's the gene we want, I don't have to go into a human's pancreas to get the cells that make insulin. Remember, every cell of your body has every gene in it. So if I want the human insulin gene, I could just do a little cheek swab and get some cheek cells, because they will have the insulin gene in it. Not turned on, but that doesn't matter. The DNA's there. So what we can do is get any cell. It could be cheek cells. It could be blood cells, not red blood cells. They don't have a nucleus. But it could also be, as we're going to see, other techniques for unborn babies. if we want to do any testing of DNA, there are ways to actually get cells from a child that is developing in the uterus before it's born. So there are lots of different ways to get the cells. Once we get the cells, what we then have to do is extract the DNA. So we've got the cells, the tissue, and there's the DNA in the cells. Now, to extract the DNA out of the cells is a very simple process, and some of you may have done this in year 10. You put the DNA from not a cell, from lots of cells into a test tube. Sorry, you put the cells in the test tube and you just use basically salt and ethanol to break open the cells, membranes and that. And so there is a way to very simply extract the DNA out of cells. So when you've got that, you've now got a test tube of lots of copies of the DNA of that cell. And every cell has 2 copies of every gene. So in my test tube, I have hundreds of copies of my insulin gene in that test tube, but I've got everything else as well, and I don't want everything else. So what we have to do is getting a way of getting our little insulin gene out of the rest of the mass of DNA. We're going to meet lots of enzymes in this story. So what we've got here is key enzymes that are involved in the tools. There's the endonucleases, which are often referred to as restriction enzymes. They're part of our story. There's a DNA ligase, which is also part of the story of recombinant technology. And there's DNA polymerase, which is an enzyme that's going to be involved in one of our tools called the PCR. So this is just a summary of the enzymes, the key enzymes that you need to know as we go through this tools and techniques part of DNA manipulation. But we'll meet each one in context as we go through it. All right, so we've got our test tube of DNA. There's a test tube, and we've got all that extracted DNA in there. And somewhere in amongst it are our genes. What we have to do, if we've got DNA, remember DNA is long stretches, we want our gene, just our gene. This is the target gene. For example, this is our insulin gene. It's just part of a DNA molecule. We just want that bit. We don't want all the rest, and we don't want all the other chromosomes. We just want our insulin gene. So if you have a long band of DNA and you just want a little section of it, you cut it out. And how they've worked out you can cut DNA is using the first of our key enzymes, our restriction enzymes. Now, what are restriction enzymes? Well, they are enzymes that cut DNA. Simple as that. They're usually found in bacteria. And only bacteria have restriction enzymes. But I want to cut out a human DNA. But remember, DNA is universal. So if restriction enzymes cut bacteria DNA, they'll cut any DNA. And scientists, and the scientists that worked this out actually won the Nobel Prize, that they now have these enzymes that are able to cut DNA. So using restriction enzymes to cut each end of the gene is a way of cutting our DNA into pieces. And one of them is our gene. But there isn't just one restriction enzyme. I think they've worked out and isolated about 4,000 by now, and it may be even more. Because what they realize is that each restriction enzyme cuts at a specific base sequence called the recognition site. So what we have to do is use the correct restriction enzyme to cut the ends of the gene. We don't want to use a restriction enzyme that cuts through the gene. That would destroy it. So to do this technology, scientists need to know what sequences are at the end of the gene. They need to know the base sequence of the gene. And scientists know that, and they know that for the human genome. They know that for a lot of genomes because they've done human genome sequencing. So by knowing the sequence of the human genomes and hence the gene and what the ends are, they can find the correct restriction enzyme that will cut the ends. Now, this restriction enzyme story, each one has its own particular sequence. And down here, we've got 3 restriction enzymes. They've got strange names. The name is after the bacteria they're found in. So the first one, EcoR1, is probably one of the first isolated. That comes from a bacteria called E. coli. EcoR1, that's the name of the restriction enzyme. What that restriction enzyme will do, it'll scan along the DNA molecule, and when it sees this sequence, it will cut. And when it sees that sequence, it will cut between the guanine and the adenine on the DNA. EcoR1 always will see that sequence and will always cut there, never anything else. The next one, HAE3, different bacteria, different restriction enzyme. It will scan a DNA sequence and it will look for guanine, guanine, cytosine, cytosine. This is on one side of the ladder. It's usually on the 5' to 3' direction. When it sees GGCC, it will cut between the G and the C. Now, you don't have to remember all these. You would be given this information. But what we've got is this specific recognition sequence and specific cutting site. And our third one, BAM, it looks for this sequence and it cuts between the 2 guanines. So it's going to cut like that. So restriction enzymes are specific to the sequence they're going to recognize and where they're going to cut, which is why you have to choose the correct restriction enzymes to cut our gene out. Now, there are 2 ways restriction enzymes can cut. For example, EcoR1, as I said, it cuts between the guanine and adenine, which on the other side of the ladder will be like that. See how the cutting's off center? It's not straight across. It's not like a pair of scissors cut straight across the DNA. When the cuts happen on the sugar phosphate backbone, they're cutting the phosphodiester bond, the hydrogen bonds between the other bases also break. So what you get is this often referred to as zigzag or diagonal cut. This diagonal cutting like this is referred to as a sticky end cut. This is actually quite a good cut to use if you can. I mean, it depends on your gene, because it allows for a bit of overlap when you're doing the next step in the technology. So it's very handy to have sticky end cuts. These actually will produce strong bonding with other DNA, which is what's going to happen down the track because you have this overlap. But in the case of Hindi, it doesn't cut diagonally. It cuts between the guanine and cytosine there and directly across. So we've got these direct vertical cuts. These are called blunt end cuts. And you can't alter Hindi to become a sticky end. It is what it is. So just understanding the first of our key enzymes in the DNA manipulation story is the enzyme to cut the gene out of the DNA. And again, to choose the correct restriction enzyme means that you need to know the ends of your gene, you need to know the sequence of your gene so you don't cut it there, and then you have to find a restriction enzyme that matches those sequences. Fortunately, those sequences tend to be very common in DNA as far as cutting of enzymes and genes and things like that. So the whole idea of restriction enzymes as a way of cutting our gene, our target gene, out of the DNA.